All right, so let's look at part 2 of this microbial world in you. All right, we're going to start here with the history. When we talk about the history of microbiology, we're going to start Robert Hooke. Robert Hooke did most of his work in the mid 1660s, and he observed and coined the term cell. So he began by looking under the microscope specifically at cork cells. And when he was looking at these cork cells, he thought, oh, they look like little rooms. He coined that term cell. Now, this marks the beginning of what we call the cell theory in biology. The cell theory in biology was completely kind of developed in 1869, but the first part of the cell theory says all living things are made of cells, which this begins here with Robert Hooke. We also see another part of the cell theory says cells are the basic structural and functional unit of life. This is why in order for something to be a living thing, it has to have at least one cell. And the last part of the cell theory is life begets life. This means that cells are capable of reproduction and cells have to come from previously existing cells. The next individual in this whole history of microbiology is Leeuwenhoek. Leeuwenhoek did a lot of his work in the late 1670s, and he kind of was a pioneer with the microscope. He was known for developing and improving the microscope by adding multiple magnifying lenses. And because of this, he was able to observe microorganisms that were actually alive. Cork cells were dead. So in this case, he was looking at live microorganisms, and he was looking through like pond water and things like that. And there were these little things moving around that he thought looked like little animals. So he called them animalcules. During this time, too, in the 1660s, we see that there was this big debate between spontaneous generation, where things just would spontaneously come out of nothing, like frogs would just spontaneously come out of ponds, or there would just be spontaneously flies coming off of rotting meat, versus biogenesis, saying that there was a biological factor behind it. So this first was studied here by Reddy, and he put together an experiment in the mid 1660s, and he experimented with maggots and decaying meat. So he was saying, hey, that maggots don't just appear on meat. There's got to be a reason why they appear on meat. And so what he did is he had a jar first with rotting meat in it, and he left it open. And he saw that flies had access to this meat, which means those flies were laying eggs on the meat, and then the maggots were hatching from those eggs. Those maggots then matured into flies. On the other hand, he had another jar that had gauze covered. And so this allowed air to move in between the meat and the jar and all that, but it did not allow the flies to have access. So no maggots appeared on that meat. The last jar, he put an actual lid on it. And again, the flies didn't have access, so no maggots were found there. So this leads to the whole idea of life has to beget life. Okay, and that life comes from those flies laying eggs, which then turned into the maggots, which then became the flies. Another thing with this spontaneous generation and biogenesis was Virchow, and this is in the 1850s. The debate was still going on. He was a major contributor to the cell theory of life begets life, but he failed to present convincing evidence to the scientific community. They did not really believe the evidence that he was giving. But in the 1860s, Pasteur helped with that evidence. His work also supported the whole philosophy behind biogenesis. Now with Pasteur, he went through the process of developing different kinds of flasks and kind of messing with the opening and how they were available to move air and molecules in and out of them. And he would use these different flasks. He would then heat them up to kill the bacteria in the fluid that was present. And then he found that some of them, if they had very easy access where microorganisms could get in, bacteria would start growing in them again. But in the one that was more convoluted, he found that there was a prevention of the microbes getting in, and so no microbial growth would occur. All right, so because of this too, he also contributed to the whole idea of aseptic techniques. And aseptic techniques are techniques where we prevent contamination from happening, where we can move one thing to another but prevent contamination from occurring. Now, this led into the golden age of microbiology. The golden age of microbiology was between 1857 to 1911. And Pasteur was right in the middle of this and that whole idea of the 1860s. Now, he was asked to kind of solve a problem. Now, fermentation was something or a process that had been going on for a long time that they used to make beer and wine. Okay, and this means that it also helped produce money because beer and wine were important for industry. The issue, though, that these individuals were having that were making beer and wine is that some of the bacteria that got into their beer and wine actually converted the alcohol to vinegar. This means it spoiled it, so it no longer could be used. So what did Pasteur do? Well, he was called on to try to come up with a solution to help prevent this. And his solution is what we call pasteurization. Pasteurization is where we use just enough heat to kill off the harmful bacteria that causes the spoiling to take place, but it can leave behind the bacteria that actually causes the beer and wine to form. Okay, if it's using the bacteria in that process. All right, so it helps get rid of the bad bacteria in order to prevent spoilage. We also see pasteurization is used for milk. Also in this golden age, we see that there was this germ theory of disease that came out. Before this point, individuals thought that diseases were caused by a type of punishment, that you were being punished for something that you had done, a transgression, a sin, something like that. However, during this time, we started to see that there was a shift that said, no, these diseases are actually linked to microorganisms, and these microorganisms got the term of germs. Semmelweis was one of the first ones in the 1840s. He was looking at what we call puerperal sepsis or childbirth fever. This is also known as a type of postpartum infection. He saw that women in certain areas were getting sick after they gave birth and with a fever, and they were dying. And this was linked to physicians who were not washing their hands. They weren't washing their hands well between each patient. And so they were transferring disease from one to the next and so on of these idea of germs. And so this is where he really started to push the idea of, hey, with your patients, you should really make sure you're washing your hands between them. Well, this is something you're like, well, duh, we do this all the time. But this time they did not know that. And so this was that development first of that whole hand washing to help prevent the movement of diseases. Lister also helped in 1860 because he was the first one to actually do an antiseptic surgery. He used a chemical called phenol, and we'll talk about phenol a little later, and it's a product that he used to disinfect surgical wounds. When he used this product to disinfect the surgical wounds, he saw that he had a reduced infection rate, which also caused a reduced death rate. Okay, so again, he's the first one to actually start kind of sterilizing in that sense of using a type of product to disinfect the individual and potentially the utensils or instruments that they were using during surgeries. Koch was also a really big player in the golden age. In the 1870s, he came up with some postulates. Now, these postulates are actually all related to linking the cause of an infectious disease to a specific microbe. All right, so you need to take a look at these postulates, and we'll talk a little bit more about them later. But his whole study revolved around this kind of concept. So if you'll see in the picture here, you have a diseased organism. Okay, in this case, this mouse, it died. And so what he would do is he would take a sample. He took a sample, in this case, of its blood, and he found some agent in the blood that he didn't think belonged, the suspected agent that caused the disease. And he would culture this causative agent on some media. Once it grew out, he would take that and then he would inject it into a healthy mouse and see what happened. In a lot of cases, the mouse died, but he wanted to make sure that the mouse actually died from what he had put into them, the causative agent. And so he would take another sample and grow it out and then do a comparison to see if they were the same. Now, there are some limitations to this, though, when we talk about this process, what Koch used. Some organisms cannot be grown out on media like we see here. One of the examples is syphilis. We're not able to grow it out. Another one is leprosy. All right, we can't grow it out on a media, and so it makes it harder for us to potentially be able to do this. Sometimes more than one type of organism can cause the same signs and symptoms. Like in a UTI, a urinary tract infection. With a urinary tract infection, it could be caused by a number of different things. And so we sometimes will have to take a urine sample and culture it out to see exactly what's causing it. Another one is that one particular organism may actually cause multiple signs and symptoms depending on where it's located. One microorganism might cause a certain set of signs and symptoms if it's found in your respiratory system versus your digestive system versus your blood. So it depends on where it's located and what signs and symptoms are there, but it's still causing disease in different ways, but it's the same microorganism. And so there are some limitations, but we do now know about these limitations. The next one we want to look at is Jenner. Jenner did most of his studies in the 1790s, and he was the first one to come up with a successful vaccine. Vaccinations help protect us against diseases. They stimulate our immune system to provide some artificial but active immunity. Now we say artificial because the individual wasn't naturally exposed. They were exposed through a syringe or a needle, okay? But they still made active antibodies against it, which also means they have memory cells to remember it later. This gives them a type of immunity. Now, the way he did this is he used cowpox virus to help protect against smallpox. So he helped come up with the smallpox vaccination. Now with this, he noticed that some individuals didn't get smallpox. These individuals were milkmaids. These milkmaids would get a similar type of disease to smallpox, which was not as life-threatening, called cowpox on their hands. And this came from the udders of them actually milking the cows. So he took their hands and he scraped their sores and he cultured out what was in there and he made a vaccine. Well, how did he test this vaccine? Well, he injected his infant son. Then he exposed his infant son to smallpox to see if he'd get it. Well, thank goodness he did not get it. Okay, this is why there's some regulations behind how we test vaccines and stuff. Okay, because that was not ethical what he did, but he did discover how to make a vaccine and how to make one successfully against smallpox. All right, the next thing we want to look at besides the history is what we call chemotherapy. Now guys, chemotherapy, what do you first think of when you think of chemotherapy? What comes to mind? Well, cancer. We think about it's a treatment for cancer. But guys, in reality, chemotherapy is just the use of chemicals to treat any disease. Now, cancer is one of those, and it's the main one that we talk about with chemotherapy. But technically, if you're taking any kind of medication for a disease that's helping treat it, you are undergoing chemotherapy. Now, there are some natural chemicals we use, such as antibiotics and things like that, to help with treatments, but there's also some synthetic drugs that are more manufactured or man-made that we use as well. Now, in order for drugs to actually be effective, they have to also be selective, meaning we have to make sure that they are pinpointing and hitting that germ that causes the disease. We don't want it to also hurt us and our cells as well. Ehrlich came up with one of these big types of chemotherapy treatments in the 1910s. He came out with what we call salvarsan. This was what they termed as the magic bullet. This was going to cure and treat syphilis and get rid of it. And it did help treat syphilis. The problem, though, is it had lots of side effects because it contained arsenic, which we know is detrimental to us. We also see Fleming in the 1920s, he developed penicillin. Now, of course, he did this on accident. He was culturing out some bacteria on some plates, and he found that some fungus was growing on it. This fungus was preventing the growth of the bacteria, and that is how he discovered it. It was giving off a chemical that inhibited and killed the bacteria. And so because of that, that's how penicillin was discovered. Again, they kind of thought it was a magic bullet too, okay? But it doesn't help and treat and cure everything that deals with bacteria. Some problems with this, especially with penicillin, is we've actually created a lot of antibiotic resistant strands. These have emerged over time due to the misuse of medications. Some of this misuse deals with the fact that maybe you've been prescribed an antibiotic and you didn't finish it. I mean, think about it. How many of us started the antibiotic, we started feeling better, and we didn't finish taking it? We helped contribute to this. On the other hand, doctors also were over-prescribing this. They were saying, oh, you're sick, here's an antibiotic. You're sick, here's an antibiotic. Well, antibiotics only help treat bacterial infections, not viruses, not other parasites and things like that. And so now we need to be more, again, selective of how we give out antibiotics, because we have created a lot of these antibiotic resistant strands because bacteria, viruses, all these different organisms that have DNA or RNA, all these different structures that have DNA and RNA, they can mutate and change, which means that their drugs may not be as effective. We also want to look here at the fields within microbiology. When we look at microbiology, this is kind of an umbrella, and there's a lot of subfields that are underneath it. One of these subfields is bacteriology, and bacteriology is the study of bacteria specifically. All right, so if you find as you're going through this and bacteria is really what's grabbing your attention, then this might be a subfield for you. On the other hand, we also have what we call mycology. This is the study of fungus. Parasitology is the study of parasites, specifically those that are found in the protozoa and the helminth groups, which remember the helminths are the worms. Immunology is also kind of underneath microbiology because it's the study of immunity, and our immunity is to fight most of those pathogenic microorganisms that we come in contact with. Virology is specifically the study of viruses. We also see that there's a new subfield based on the idea of DNA technology. This is known as recombinant DNA technology, which is known as gene manipulation. Manipulating genes in order to be able to study these changes and mutations that take place, but also where we manipulate the organism. One example is here, like how many of you know a diabetic? Okay, and that diabetic is insulin dependent. Well, where do they get their insulin? Have you ever been asked to donate insulin? No, because the insulin you make, you need. Before we actually had insulin in the pharmacy in the way we do now, you could get insulin from cows. And cow insulin worked, but it wasn't as good as human insulin because there were some differences. However, the insulin now that you buy at the pharmacy is human insulin. Well, how do we make it? Because you don't donate it. Well, we make it through recombinant DNA technology. We take the gene, because we know what gene makes insulin in the human body, and we take it out and we put it into a bacteria. We then manipulate the bacteria to make that insulin. Well, the bacteria doesn't need insulin because it doesn't have blood and it doesn't have blood glucose. So we're then able to harvest that insulin out of the bacteria and package it and be able to give it to individuals who have diabetes. Now, this isn't the only thing that we use that for. Other hormones are used as well as different antibiotics. There's a lot of different things that we use this recombinant DNA technology for. And it is kind of a sub area of microbiology because we use normally microorganisms to do this. Now, when we look at microorganisms, we also want to see how they relate to human welfare. How are they beneficial to us? I mean, because that's kind of the bottom line most of the time. We're very selfish in that sense of like, how does it benefit us? Well, on the ecosystem side, the ecological benefit of microorganisms are going to be that some of them recycle vital elements within our ecosystem. Those decomposers that are going to decompose dead, decaying material, they are going to help return certain elements to the soil, to the ecosystem. This includes carbon, nitrogen, oxygen, phosphorus, and sulfur, which you can see here with those letters. We also see that microorganisms are used in sewage treatment plants. This is to help reclaim water and to clean the water so that we can continue to use it, using microorganisms to do so. GMOs are used to clean up pollutants. GMOs, guys, are genetically modified organisms. They've modified certain bacteria to where they actually want to eat toxic waste. They want to help clean up chemical and oil spills. They take in those chemicals and they neutralize them. We also see that they can provide safer ways for us to control agriculture, and they do this by certain insect pesticides. Instead of spraying them with chemicals, we're using some of these microorganisms to help with this. Another thing, though, is how do they medically or biologically benefit us? Well, one, some of them produce antibiotics that we utilize against bacteria. Others are used in gene therapy research, where we're looking at how genes work and how we can manipulate and move them not only from bacteria and stuff like that, but also how can we deliver them into the person who needs that new gene. And viruses are one way they look at that. We also see that recombinant DNA that we talked about a minute ago, making vaccines, insulin, growth hormone, different enzymes you might need. They also can help us generate hardier crops, where those crops can live in drought conditions or bigger temperature changes, or even be insect resistant. They can also improve food products. We call those super fruits or vegetables. This is where they can actually be larger, or they also have a better shelf life so they don't go bad as quickly. On a biological sense as well, you have what we call normal microbiota, or your normal flora that's on your body and in your body. They are going to help prevent invasion of pathogenic organisms. They're also going to help supply useful vitamins for you. This is a symbiotic relationship. You're helping those microorganisms and they're helping you in return. Of course, then that's a benefit. All right, well, what about the money side, the economic benefits? Well, these microorganisms, different ones, can help produce products such as alcohols, acetone, and vinegar. We also see they can help us make certain foods like sauerkraut, pickles, soy sauce, cheese, yogurt, bread, and the list could go on. They're also used in different industries like for biodiesels, also clothing products. In your textbook, it talks about stone-washed jeans, and these are actually making a fashion comeback right now. We see that there's economic disadvantages though too. We do see that some microorganisms cause our food to spoil quickly, or they can cause water contamination, which does cost a lot of money in that sense to clean up that water. Now we're going to kind of go back in a circle back to that first slide we talked about in chapter 1, where we talked about being pathogenic. Remember, pathogenic means that it is an organism that causes disease. Infectious diseases are caused by things that are microorganisms. They're caused by bacteria, viruses, fungi, or protists. Now, these can be passed on from person to person. An example of this is diphtheria, where they actually get passed on through respiratory droplets from person to person. They could be transmitted through bites, like insect bites or animal bites. So mosquitoes, a lot of times, they get that bad rap, but they do carry a lot of different things like malaria, West Nile, things like that. Ticks, they carry Lyme disease, Rocky Mountain spotted fever. We'll talk about a lot of these throughout this course. Okay. Also, you could acquire them through environmental exposure. An example of this is cholera. Cholera you can get through drinking contaminated water. All right, so you could be exposed in the environment to it. Now, one thing that's important when we're looking at infectious diseases is that a lot of times there's kind of patterns that come into play, or there's new ones that kind of emerge and become a problem. Emerging infectious diseases are new or changing diseases that are increasing, or they have a potential to increase in their incidence. So some virus examples of this are SARS, H1N1, which is a type of influenza, avian flu. They're all ones that we watch. We need to add to the list COVID. Okay, COVID-19 needs to be added to that list. But when we look at these guys, these are all kind of forms of a coronavirus where changes have occurred. We also see, though, there's lots of viruses like West Nile, Ebola, and HIV that need to be monitored and watched. We also see there's some purons. Purons, guys, are unique because they are even smaller than viruses. Purons are mutated proteins. And one example of this is Creutzfeldt-Jacobs disease, or CJD, and this is very similar to mad cow disease, but it affects humans. Some bacterial examples that need to be watched are MRSA. This is a type of antibiotic-resistant staphylococcus. Also VRSA, another one. It's an antibiotic resistant to another, vancomycin, a different antibiotic. We also see C. diff. You may have heard of C. diff before. It's a very contagious type of infection that affects your digestive system. E. coli is another one. Guys, E. coli is one of those normal microbiotas that, as long as they're in your intestines, your large intestines, they're helpful to you. But if E. coli gets anywhere else in your body, it can make you very sick. Your notes also talk about necrotizing fasciitis. Necrotizing fasciitis is a flesh-eating bacteria. So these are just some examples of things that the CDC, the WHO, and even other doctors they're watching. They're continuing to watch and monitor. Okay, and so it's not just, you hear a lot about certain ones, but there's a large list of these, and we're going to talk about a lot of them throughout this semester. But guys, we focus a lot of times on the whole idea of the pathogenic ones, the ones that make us sick. But are the majority of microbes actually pathogenic, or are they beneficial? Well, believe it or not, majority of them are beneficial. Okay, most of the microorganisms that are out in the world actually help us. Only a small percentage are pathogenic, but those are the ones that we like to focus in on because, again, they're the ones that make us sick, and they're the ones we need to try to stop. All right, and a lot of what we're going to talk about does revolve around those pathogenic. But we do need to remember that there are lots of microorganisms that are actually helpful to us.